Hello, everyone, and welcome to today's AZO Network webinar, which is brought to you by News Medical and our sponsors for today's event, Vazyme Biotech. A big thank you to the team at Vazyme for making this event possible. My name is Danny Lazel, and I am delighted to be hosting today's webinar. Now, before getting started, we have just a few points about how to get the most out of your experience today. Firstly, the event is being recorded, and we'll be sharing the on-demand link with you within the next 24 hours. So if you enjoy what you see today and you think you have a colleague or peer who might benefit from also seeing the content, then please consider sharing with them internally or via your social media channel of choice. If you happen to experience any technical difficulties, then they are normally resolved by refreshing your browser. But if this doesn't work, then send us a message using the Q&A box and our backend team here will get you back up and running as soon as possible. Now, today's event will run as three parts. First, we are joined by Qiming Zhang, lab supervisor at Peking University and postdoc associate at MIT, who will start the webinar with the first presentation in just a moment before handing over to Shuyong Ding, technical support engineer at Vazyme. Once these two presentations are over, we will then open the floor for a Q&A session. So if you have any questions that come to mind during the event, then use the Q&A box on your right-hand side to send those in, and we'll get to as many as we can during the Q&A session after the presentations. If you happen to be watching this on demand, then you'll find the Q&A box on the bottom right of your screen, and these will be replied to via email. Finally, once today's webinar is over, we would greatly appreciate it if you could give us 2 minutes of your time to complete the end of webinar survey. This will appear on your screen once you are done today. Now, I'm delighted to welcome Qiming Zhang to get things started. Qiming, over to you whenever you're ready.
Hi everyone, my name is Qiming Zhang. I'm currently a postdoc at Koch Institute for Integrative Cancer Research at MIT. I had my PhD training in Peking University in China. It's a pleasure to be here today to share some of my research projects. And the title of the topic today is The Landscape and Dynamics of Liver Cancer and Liver Metastasized Colorectal Cancer Characterized by Single-Cell Sequencing. So here shows the outline. So I will start from background and then scientific questions and study design. And the most important part are the results and discussions. So let's get formally started from background. For liver cancer, it can be divided into primary and secondary liver cancer. For primary ones, liver cancer has the fourth most mortality in the world, and it has higher incidence and mortality rates in males than females. And liver cancer can be divided into three main subtypes, including HCC, CC, and combined HCC and CC. For secondary ones, the liver is the most common site for tumor metastases. More than 50% of colorectal cancer patients could develop liver metastases. And as we know that the immunotherapy strategies has been applied on both CRC and HCC, including checkpoint blockade and adoptive T-cell transfer. Especially for checkpoint blockade, anti-PD-1 and anti-PD-L1 has been applied on HCC. However, if we see the single agent, the efficacy is not so well on HCC. But if we do combine therapies, for example, if we combine anti-angiogenesis VEGF-A and anti-PD-L1, the response rate will be much higher. In addition, if a patient has liver metastases, the prognosis will be worse and also the drug response will be lower. And this is an example based on melanoma patients and lung patients. So this phenomenon showed that maybe there's more populations in the tumor or in the tumor microenvironment. That's why the response in HCC or in liver metastasis is not so well. And actually, if we dissect in the tumor microenvironment, there is not only cancer cells, but also other cell populations, including immune cells, for example, lymphoid lineages like T, B, NK cells, or myeloid cells, including macrophages, dendritic cells, and so on. And also, there are stromal populations like fibroblasts, endothelials, and so on. So to dissect these different populations individually, the single-cell technologies has been a powerful tool to study the complexity of the tumor microenvironment. The single-cell technologies has been invented in 2009, and during these few decades, there are multiple snRNA-seq technologies coming up. And mainly it can be divided into two different ways, which is plate-based, is full-length, and it's usually we need to do it manually. It's very laborious, but you can have the full length. So it means for individual cells, you would have more RNA information. And also there's droplet-based technologies, for example, 10x genomics. It's usually you only have the RNA-seq information from 3' or 5'. It's not full length, but it's highly throughput. So you can have multiple cells in one batch of experiment. In my project, I actually combined these two different technologies and compared them to see which way is better. In addition, if we see the single-cell RNA-seq studies on liver cancer, we can see in the recent years there are multiple studies coming up. This example shows the functional states and transitional relationships of different T-cell populations in HCC. And in this paper, the authors divided liver cancer patients based on the tumor transcriptional biodiversity, and they divided them into two groups to compare the TME difference and how they are related to the patient outcome. What's more, we can see that there are some studies, they study not only primary HCC, but also early relapse HCC to compare the TME difference of those patients. And besides, for liver metastases, there are also some studies. They show that both intrinsic and extrinsic factors are associated with liver metastases. So the last paper, they are talking about the intrinsic factors. We can see that they are talking about different mutations are related to the expression of TGF-beta, which drives immune evasion. And for the right paper, they are talking about how the liver microenvironment, the hepatocytes, would directly form the pro-metastatic niche and how they can influence the primary tumor. Even though there are several studies, there are still several unresolved scientific questions. And here I listed some, and also based on these scientific questions, we designed our study. So the scientific questions mainly include basic ones, mechanism questions, and also clinical questions. For basic questions, it's very simple. It's just what the exact cell populations within liver cancer. Even though the question is simple, still we are not currently we don't have the full landscape of liver cancer. So it's still an unresolved question. And for mechanism questions, that's what are the relationships of these different populations, and especially for liver metastases, what's the relationship of metastatic sites with primary ones? And also clinical questions, they are mainly how to apply those scientific findings into patients. So how to find important cell populations, how to find biomarkers, and so on. To resolve these questions, we actually directly collect clinical sample. So I collected tens of patients with primary liver cancer or colorectal cancer liver metastases. There are multiple tissues, including the tumor sites, adjacent liver or adjacent colon, and also blood, lymph nodes, ascites, and so on. So based on these clinical samples, we performed single-cell RNA-seq experiments. We used both 10x and SMART-seq2. And before the single-cell RNA experiment, we mainly use flow cytometry to collect immune populations, use CD45. So after we have all the data, we will then integrate all the data and do a series of single-cell analysis So the first step is to use unsupervised clustering to do cell identification. After we have the identity of each cells, we will compare the cell identity in different tissues, so that we can see which population is enriched in tumor, which population is enriched in adjacent normal, which in blood, and so on. And after that, we will dig in the relationship between different cell populations. These can be divided into two parts. The first one is trajectory analysis, so that we can see which population is the origin of the important populations. And also, we will do a cell-cell interaction analysis, which is to see how the cancer cells are regulated by different populations and how the immune cells, for example, T cells, are regulated by different populations. And after that, since we have different tumor sites, we also do some cross-cancer type comparisons. So for the cell trajectory and cell-cell interaction analysis, I listed two tools here. So for trajectory analysis, we mainly use RNA velocity, which utilize the splicing and unsplicing rate ratio to determine which cell population is the early stage, which is the later stage. And for cell-cell interaction, we mainly use the cell phone DB tool, which utilized the expression level of ligand and receptor of each cell, and they integrated multiple information based on literature and other resources. And based on the expression of ligand and receptor, they can infer whether these two populations are interact with each other or not. Okay, so let's now move on to the results part. First, after we collected around 20 primary liver patients and liver metastatic colorectal cancer patients, the first thing is to identify the cell identity of each cells. So the left Hue map shows the cell landscape of primary liver cancer. So we can see that the major cell populations are all included, including these are lymphocytes. There are TB, NK cells, plasma B cells, and so on. And also there are myeloid cell populations. And the red panel is cell populations in CRLM patients, which is colorectal cancer liver metastasis patients. So also we can see similar populations here, so which is actually similar compared with HCC patients. So now let's focus on the myeloid part. If we zoom into the myeloid population, we can see there are also different populations. Each color represents one cluster, and we can see dendritic cell populations, monocytes, macrophages. Especially for dendritic cell populations, which are here, we can identify the conventional DC1 and DC2 cell populations. In addition, we also see unique population which highly express a list of genes which represents the dendritic cell maturation and migration, including CCR7, CCL19, LAMP3, CD80, and so on. So if we use a trajectory analysis to see how these mature population are associated with the previous reported conventional DC1 and DC2 populations, we can see that the trajectory flow is that from both CDC1 and CDC2, they are pointing to the mature population, which means maybe during the dendritic maturation, the cell they lose their original cell identity marker, and then they express similar genes that are associated with maturation or migration. Then we want to validate this phenomenon at the protein level. So we use flow cytometry, and we use different markers to compare whether these populations are different in different tissue types. And we can see that in the tumor tissue, we can see the most LAMP3 populations in tumor, and compared with adjacent liver tissue or negative control, and the quantitative results are here. So in addition, if we also divided CDC1 and CDC2, we can see that the maturation and migration marker CD80 and TCR7, they showed the highest expression in LAMP3 DC populations compared with DC1 or DC2. That means these results at the protein level are consistent with what we observed based on snRNA-seq, which is transcriptional level. And also, we performed in vitro validation to see whether different dendritic cell populations could all become LAMP3 disease after different stimuli. So we used primary disease from PBMC, and we used different stimuli, which had two conditions, including LPS plus interferon gamma and poly-IC. So if we see these different groups, we can see that actually in different dendritic cell populations, including DC1, DC2, or plasma disease, in different stimuli condition, the LAMP3 and also CD83 are all goes up. So which means these LAMP3 are a common marker of different dendritic cell population maturation. And also, we performed single cell RNA-seq on this in vitro population to see that if we can actually recapitulate the transitional flow of these different populations. So based on this UMAP, we also did RNA velocity, and we can see that each cell has an arrow, and the general flow is that the different dendritic cell populations are all point to the LAMP3 dendritic cells. So this means that the in vitro results we observed is consistent with the in vivo results we observed in patients. That means maybe in HCC patients, we saw that LAMP3 dendritic cells are enriched in a tumor and also in lymph node, which means that maybe the LAMP3 disease in lymph node, they are activated by tumor-associated antigens, and then they are mature and they can migrate into the tumor, and maybe they can prime T cells and or NK cells, and then the T or NK cells can exert their tumor killing functions. In that case, those dendritic cells should interact tightly with different lymphocyte populations. So that's why we perform cell-cell interaction analysis. So based on this analysis, we generated an interaction network, and we can see that those LAMP3 dendritic population are actually interacting with different CD4 and CD8 populations, including the exhausted CD8 T cells, Tregs, and also Th1 populations. We also validate these results at the protein level. So from the multicolor immunohistochemistry results, we can see that in this picture, there are CD8 T cells, they express PD-1, and CD80 dendritic cells, they express PD-L1, and they are actually physical adjacent with each other. So that means maybe they are now interacting with each other. So this is the primary HCC part, and this except for that, since we have both SMART-SIG2 data and 10x data, we also compared whether these two different technologies can contribute to the dataset. So as we talked before, the SMART-SIG2 is laborious. We need to do these different cells one by one. So the cell number of SMART-SIG2 is much less than 10x. However, the 10x data, they are high throughput, but their detected gene number are much lower than SMART-SIG2. So if we separate these two datasets and then do the clustering individually, we can see LAMP3 dendritic population shows up in SMART-2 data, but not in the next dataset. So that's why we need to integrate these two datasets. And in the integrated ones, we can rescue those populations that might miss in these two different platforms individually. And this shows the gene number. So if we take out this LAMP3 dendritic population and to compare different biological process of geoterms, we can see that different biological process, the detected gene number of SMART-2 are much higher than 10x. So this means that incorporation of SMART-2 data into the widely used 10x-based RNA-seq study has synergistic effect to identify immune cell populations at a higher resolution. And then for the other study, maybe we can consider use SMART-2 as a small population, but they have more genes, and then use 10x to have higher cell numbers, and then to integrate different data sets together. And those results has been published in 2019. And after that, since we have primary HCC data, so we are considering about how the liver metastasis looks like. And also based on the paper, they include primary CRC patients. They mainly talking about different myeloid populations, especially two tumor-associated macrophage populations. In their study, they identified two different TAM populations, which including C1QC TAM populations. They are more antigen presenting, and also there's one SPP1 TAM population. Their function seems to be more pro-tumor, including tumor angiogenesis, metastatic liver cancer, and so on. So based on our previous primary HCC data and this published primary CRC patients data, we want to compare how the microenvironment, the similarity and difference of different tumor microenvironment of different cancer types. So if we see these two macrophage populations, we can see that for C1QC TAM, we can see in both in HCC tumor and in CRC tumor, it shows high presence. However, if we see the SPP1 population, we can see that it shows high presence in CRC tumor, but in HCC, the fraction is very low, and it's similar to adjacent normal tissues. So which means this SPP1 TAM population is specific in CRC tumors compared with HCC. So what it looks like in CRLM patients. So if a patient with liver metastasis, rather this SPP1 macrophage population would exist in the liver. And this UMAP shows the macrophage and monocyte populations. And if we see the heat map, including different populations in different tissue types, we can see that actually this is the liver metastasis, which is metastatic liver. Both C1QC TAM and SPP1 TAM are highly present in this tissue. So which means in primary HCC, there is no SPP1 macrophage, but in metastatic liver tumor, there's a lot of SPP1 macrophages in it. So we next developed an algorithm to see what's the relationship of the cell populations in metastatic sites and how they are similar to different other tissues. For example, how they are linked to primary tumor, to lymph nodes, to adjacent colon, adjacent liver, and peripheral blood. So based on this algorithm, we can get these results. So we can actually link each population in metastatic tumor to their potential origin of different other organs. And based on these results, we actually see that both C1QC TAM and SPP1 TAM in liver mets, they are actually linked to the primary tumor. Also, we have some lymph nodes metastatic information. If the lymph nodes are actually tumor associated, if it's metastatic lymph node, then we can observe several SPP1 macrophages. But if the lymph node is not metastatic ones, then there's no SPP1 macrophages. So now we can conclude that this SPP1 macrophage population, they are associated with metastasis, which means they are tightly linked to the malignancy instead of the host organ microenvironment. So if we directly compare the tumor sites of different organs, we can see that for primary CRC and primary HCC, and also the colon tumor site of CRLM patients, they are all they are not as high as the liver metastases. And also, if we check the marker genes of different time populations, we can see that those SPP1 associated marker genes, they are highly expressed in CRLM patients and in CRC patients, but not HCC patients. So this means these SPP1 macrophages, maybe they exert pro-tumor and pro-metastatic process. So in that case, if we check the overall survival, we should see that SPP1 macrophages are associated with worse survival. So this is actually what we observed here. If we use SPP1 gene or SPP1 signatures, we can see higher presence of SPP1 means worse survival. And in addition, we also validated their fraction in primary sites and metastatic sites. And we saw that at the protein level, also the SPP1 populations in primary tumor are less than in metastatic tumor. So actually, if we use the phenylalanine algorithm, we can also link other cell populations in metastatic tumor with their populations in different other organs. And the detailed results are in this paper. This is actually published earlier this year. And these are all the results part. So let me summarize here. From the technology point of view, we compared SmartSeq2 data and 10x data, and we conclude that incorporation of SmartSeq2 data into 10x data may have synergistic effects. And for HCC, we mainly focus on the LAMP3 dendritic cells, and we saw their migration property and maturation property, and also we saw that they might interact with a lot of lymphocyte populations. And for CRLM, based on our data, we saw that SPP1 macrophages, they play an important role in the process of liver metastasis. So in addition, although we have some results, there are still several unresolved interesting questions. For example, for macrophages population, if we know that now SPP1 TAMs are associated with liver metastasis, but how they exert their functions exactly, we do not know that, and how to apply this population on patients or on the clinical guide, still we need more evolution and more information. Also, for dendritic cells, we saw these unique LAMP3 populations in liver cancer, and also there are several other studies reported these populations in other cancer types. but how they exert their functions, because we saw that they can interact with different T cell populations, with Tregs, with exhausted T cells, with Th1 cells. Are they all exert positive functions, or are there like multiple functions of this population is not clear yet. And for other interesting questions, including how the heterogeneity of myeloid cells would influence on cancer initiation, and also the metastatic part. Those are all important, interesting questions that are not resolved yet. So I will stop here, and thank you all for your attention and listening. That's all. Thank you.
Thank you, Kimi, for that fascinating presentation. Before we move on to the second part, just a quick break to take an audience poll, which should be appearing on your screen now. We're just interested to know which of the following brands do you purchase library preparation kits from? So you've got 6 options there. If you answer the one that comes to mind. And whilst you're doing that, I'll hand over to Shuyan to get things underway. Shuyan, over to you.
Thank you for joining our webinar today. I'm Shu Jing, Technical Support in Vazyme. The topic of today's webinar is NGS Library Preparation. Let's start with the introduction. Next Generation Sequencing, NGS, is also known as Massively Parallelized Sequencing. Instead of one at a time, it is able to sequence lots of DNA strands all at the same time and determine the DNA or RNA sequences to study variation or mutation in genes. This technology now has been widely used in different disease research, such as genetic disease screening, tumor detection, reproductive health screening, and public health control, etc. The NGS workflow can be divided into 4 major parts: nucleic acid extraction, library preparation, sequencing, and data analysis. Let's begin with the first step, nucleic acid extraction. Nucleic acid extraction is the first step of NGS experiments. It consists of 3 major processes: isolation, purification, and concentration. Usually, we extract DNA or RNA depending on the circumstances. After extraction, it is better to check the quality of the extracted DNA or RNA before moving on to the next step. The common instruments we use are nanodrop, qubit, and bioanalyzer. Nanodrop and qubit are commonly used to check the quantity and concentration of the extracted samples, while bioanalyzer can be used to determine the sample fragment size to see if it is degraded or not. If the quality of all extracted samples are great, then it is good to move on to library preparation step, which is one of the most important steps in NGS. We will start with DNA library preparation first. For library preparation, DNA first needs to be fragmented into smaller pieces. There are 2 different fragmentation methods: physical-based fragmentation method and enzymatic-based fragmentation method. Physical-based method requires physical sharing machine. Since physical sharing machine will randomly fragment DNA sequences, the fragmented DNA will have strong randomness. After fragmentation and repair, adapter ligation, and library amplification can be performed to build library. Vazyme DNA library preparation kits included this module and are applicable for DNA library preparation on broad DNA input range and extensive species. In addition, Kit ND608 has additional DNA damage repairing module, which is specifically designed for FFPE samples to reduce false positive rate and increase sequencing quality. If physical shearing machine is not available, we could choose enzymatic-based fragmentation method. This method fragments DNA sequence by enzyme mix, so the sample wastage is low. Therefore, it is recommended when the samples are rare or precious. Vazyme enzymatic-based DNA library preparation kit has the advantages in combining defragmentation and repair and de-entanling 3 steps into one single step, efficiently simplifies the experiment procedures, and saves a lot of time. Move on to RNA library preparation. Since total RNA we extracted from our samples contain large percentage of ribosomal RNA, these ribosomal RNA are very constant. So usually we do not want to analyze too much on these. The RNA we are actually interested in are usually mRNA, which only take up 5% of the total RNA. So RNA enrichment step is an essential step in RNA library preparation to get rid of undesired type of RNA. To do this, we have 2 strategies. First, by using mRNA capture kit, we can capture mRNA poly-A tail by beads. This strategy is fast and easy, and could only use for RNA samples with poly-A tail, mostly eukaryotes. The second strategy is ribosomal RNA depletion. Basically, use probes to capture ribosomal RNA and get rid of them. This strategy can be used for total RNA sequencing for both eukaryotes and prokaryotes. And Vazyme designed various ribosomal RNA depletion kits for different species and sample types. After RNA enrichment, we can perform RNA library preparation. Since RNA are much unstable compared to DNA, fragmentation of RNA can be performed by enzyme, heat-alone, or exposure to cations. Vazyme RNA library preparation kit fragments RNA into small pieces by incubation with magnesium ions at 94 degrees. It combines the cDNA second-strand synthesis and repair and de-entangling 3 steps into 1 step, which is very time-saving. Purification and site selection are also important to perform during library preparation to get the desired library for sequencing. The use of clean beads is one of the easiest and fastest way. DNA clean beads are commonly used for DNA or RNA library purification and library site selection, while RNA clean beads could use to concentrate RNA libraries in low concentration, or purified ribosomal depleted RNA in the RNA enrichment step. The last step before sequencing is library quality control. qBit and qPCR can be used for library concentration detection and quantification to check if the amount of prepared library is enough for sequencing. Since qBit measurement is quick and easy, it is the most frequently used library quantification method. qPCR method could perform absolute quantification by identifying the P5 and P7 sequences in Illumina platform libraries. Therefore, if a more precise measurement is needed, or the experiment is still in the troubleshooting mode, qPCR method will be recommended. Bioanalyzer is another use for library quality control instrument. By using this, library fragment size could be checked to see if the majority of fragment size are within the desired range. In this stage, Vazyme could provide total solutions or reagent for qubit and qPCR library quantification. If all libraries meet the sequencing requirement, then they will be pulled onto sequencer for sequencing. Currently, the most widely used NGS sequencer are Illumina, MGI, and Ion Torrent sequencing platform. And we are able to provide library preparation kit, modules, and reagents support all these platforms. Here's all for today. Thank you.
Okay, thank you for those two presentations. We are now going to move into the Q&A section of today's webinar. Just before we do, just want to draw everyone's attention to the handout section, which you can find on the right-hand side of your screen. In this handout section, you're going to find some additional resources that can supplement what you've heard today, and also, so some good content there. So please do check those out and download those whenever you're ready. We've had lots of good questions come in, some of which have been answered during the course of the presentation. So we'll move on to this first one, Christina, which is for you.
Yes.
So if the RNA residue is still relatively high after mRNA capture, is it possible to further reduce it?
Since the capture bead's binding affinity is varied on different species, some species may result in relatively high ribosomal RNA residue after mRNA capture. In these cases, we suggest to perform a second round mRNA capture to further reduce the ribosomal RNA residue. Also, if the RNA sample shows degradation, for example, the ring value is low, then it will also affect the mRNA capture efficiency. For these situations, we suggest use ribosomal RNA depletion method instead of mRNA capture.
There's another question here about size selection. So if size selection is an important step during library preparation to control the distribution of the insert, then which is the most appropriate stage in the experiment to perform size selection?
And actually, Vazyme DNA or RNA Library Preparation Kit is applicable for site selection at various stages before and preparation, after adapter ligation, or after library amplification. So site selection after adapter ligation is usually recommended to most of the cases. However, if the input samples are sufficient but with low purity, then site selection before and preparation will be recommended. On the other hand, if the input DNA amount is low, to reduce the loss of input DNA during the workflow, it will be better to perform site selection after library amplification.
Kimi, the first question. It seems that LAMP3 plus DCs can interact with multiple cell types, including the exhausted T cells, the Tregs and Th1 cells. What are the exact functions of LAMP3 plus DCs? Are they anti or pro-tumor?
Yes, thank you for this question. This is actually a very interesting and important question because we can see that if LAMP3DC can interact with exhausted T cells or Th1 cells, maybe they are anti-tumor, but they are also interact with Treg cells, so which means also they are pro-tumor. So maybe this population has a dual function in the tumor microenvironment, and actually. an easy way to test is that we use the survival analysis to see whether the composition of this population in the clinical patients, they are associated with good or bad survival. Maybe they can explain their global functions. So based on our results, we saw that the LAMP3 dendritic cells is actually associated with better survival in HCC, which means maybe in liver cancer, the global function is anti-tumor. However, we also see that in some other cancer types, the LAMP3 dendritic cells are associated with worse survival. So maybe this population has different functions in different cancer types.
Perfect. Thank you for that comprehensive answer. So the next question for Qiming is, it's interesting that SPP1 plus the macrophages are present in the CRC but not HCC. How about the other cancer types?
Yeah, this is actually a related question with the last one, because we did observe different populations in different cancer types. And for SPP1 macrophages, we saw that this population exists in CRC and also in lung cancer, breast cancer, and so on. However, it doesn't exist in HCC. And also, the presence of this population is very rare in melanoma and also in stomach cancer. So an interesting phenomenon is that although in HCC or other cancer types, there's no SPP1 population, there's other population of macrophages. They exert angiogenesis function. So maybe there's some synergistic effect of those different cancer types and for this population.
Fantastic. And then the final question, how do you apply the important populations on clinical or therapeutic guidance?
Yeah, so I think the most direct way to apply the clinical or clinical guidance is to apply our data on patient samples. So we can use our single cell data to generate several gene features for different populations, and then use these features to do survival analysis. If we observe several populations, they are strongly associated with better or worse survival, that means this population is strongly pro-tumor or anti-tumor. And then we can narrow down several important populations. Within these populations, we can then select several important genes. And we can also check if these genes are associated with the currently clinical trials or candidates or any drug candidates and so on. Yeah.
Fantastic. Well, thank you very much for those answers. Thank you for your presentation. We'll bring the webinar to an end now. Thank you everyone for joining us today. And do be sure to check out the handouts before you finish leaving the webinar room today. And if you could share some of your feedback in the end of webinar survey, that would be greatly appreciated. Thank you to both of you again. Enjoy the rest of your days, everybody, and we'll see you all again soon.